Yeah, thank you. So I think one of the overarching challenges facing astrobiology and origins of life research is this need to find a set of principles that we know can apply to both detailed and general problems of living systems in a variety of contexts. And this has generally been challenging thus far, and the hope is that we can someday find this set of principles. Today I'll talk mostly about how we can go from extant life to thinking about the types of general principles that life points us towards. So I think typically there are two ends of a spectrum for thinking about the principles of life. At one end, we have a set of abstract theoretical principles, typically mathematical or computational. And these are often very general, and we should expect to apply to any living system for which we can shape them into one of these constructs. And at the other end, we have all of the life we've seen. And the question is, can we take all the life that we've seen and extract another set of principles from that as well? And the hope is that from both ends of this spectrum, we can find some set of general principles. Once you have those, you can build some universal theory of life similar to the universal theories of chemistry and physics that we already have. And then once you have this theory, you can expand back out into specific applications, such as thinking about a variety of origins of life, or thinking about searching for life in astrobiological context. So at this end of the spectrum, where we have these abstract theoretical principles, we already have some of these. It's worth noting. Examples include the error threshold or pattern formation. So for the error threshold, we know if you can formalize how a system replicates and you can formalize what the inheritance mechanism is, then you can set a maximum mutation rate in order for that system to be able to evolve. Now the trick here becomes abstracting a variety of systems into this framework, but if you can do that, we have a lot to say. So this is one nice principle that we already have. Another would be pattern formation. We understand how certain sorts of dynamical systems give rise to patterns naturally, even give rise to sort of naturally replicating systems. And so in this end of the spectrum, we have two of a very long list of principles we've already built up. And then for extant life, we have a lot of very detailed knowledge about particular physiology or what our actual genetic inheritance mechanism looks like, what the central dogma of molecular biology looks like. And so the question is, how do we take those specific details and generalize them into this set of general principles where they start to meet these abstract theoretical concepts that we already have? And so this is what I'll talk about today, is how we can look at life in a slightly different way than we typically have and start to find some generalities there. One nice example is the observation of allometric scaling or power laws in biology. So these show up for a variety of classes of organisms. And what they typically show is that if you have many orders of magnitude in organism size plotted against some feature of an organism, in this case metabolic rate, you see that all organisms within a class fall along a single power law relationship with some amount of scatter. So this is sort of amazing because it says that despite all of the detailed evolution that's occurring, adaptation to particular niches, particular evolutionary histories, path dependency, despite that, there's some set of constraints common enough to a body plan to give rise to these single regularities for diverse organisms. What's also interesting about these relationships is that they shift as you go across organism architecture. So these are all single-cell bacteria, then you shift to the single-cell eukaryotes, and then you go to small multicellular eukaryotes. And each time you do that, the exponent of this power law shifts, telling you that there's some shift in constraints associated with architecture and metabolism. So this is great, and if we can just understand what principles are causing this, we might be able to say something very general about the connection between organism architecture and various physiological features. We can also take something like this and make it much more detailed, for example, by setting up a very simple partitioning of metabolism. So if we say in general metabolism needs to be partitioned between some ability to produce new biomass and towards replication, and some amount of energy to maintain the biomass that you already have, because there are certainly decay processes, then we can say metabolism equals growth plus maintenance. We can rewrite that in a more detailed bioenergetic form, where you have metabolism on the left-hand side here is equal to some power law, and that equals some energy to produce a new unit of biomass times the rate of biomass production plus some energy to maintain an existing unit of biomass times how much mass you actually have. And then you can solve all of this to predict a variety of things, such as the growth trajectory of a single organism through its life cycle, or the population growth rate of organisms, where if we know the sort of average bioenergetic parameters of life, which we do, we can then make these broad predictions for, say, all bacteria, all unicellular eukaryotes, and all small multicellular organisms in terms of the specific population growth rate as a function of body size. And so again, we see these architectural shifts showing up here, where as bacteria become larger, they grow faster and faster, whereas the unicellular eukaryotes, as body size evolves to be larger, grow more slowly, and this is due to the difference in these energetics as connected with architecture. And we see that we can predict distinct scales where these architectures break down. So we can set a minimum size on bacteria, where maintenance becomes the entire metabolism. We can set a maximum size on unicellular eukaryotes, again, where maintenance becomes the entire metabolism. And here we see that this does a good job of predicting where you stop seeing unicellular eukaryotic life and you start seeing small multicellular eukaryotic life. So this predicts the need for an evolutionary transition. And this sets a lower bound on sort of the smallest bacteria we see, which agrees very well with the recent measurements out of Jill Banfield's lab, which has the new world record holder for smallest life. So this is really nice. We now have these broad taxonomic predictions. We can understand where architectures fail. And we can also continue to drill down on one class of organisms to try and understand something more specific about the physiology, again, from a very average or general perspective. So for example, one thing we can do is knowing what we know about the energetics of these organisms, we can start to predict the need for ribosomes at various scales. So we could take the average physiology of the central dogma and break out each component and understand what's driving that. And so in this case, I'm showing our theory for the predicted number of ribosomes converted to volume units as a function of cell size just in bacteria. This is the total cell size. This is sort of a best power law fit to those data. And here are the very messy data from a variety of bacteria, where this is messy mostly because ribosomes are sort of one of the main dimensions where organisms tune physiology. And so what we see here is we do a good job of predicting this broad consistency of organisms. We also predict an upper bound on bacterial size. Right, so we predict now that there becomes a distinct scale at which you would need more ribosomes than can fit in the cell. So this now predicts an upper bound on bacteria, which we didn't have in this relationship. But because this growth rate is increasing quickly, you need ribosomes to keep up with that, and so eventually that becomes impossible. So we have another reason for this architecture breaking down. Now I won't show it, but we've expanded this to sort of capture all of the main components of bacterial physiology. Here is total cell volume for reference, and then everything else is converted to volume units for cross comparison. And you can see that bacteria across their range have sort of this dramatic change in bulk physiology, where they go from being mostly composed of proteins and DNA encapsulated in a membrane to being mostly RNA components as the cell becomes larger. And eventually this limits their total size. So this has a variety of implications for what you might measure stoichiometrically in an environment. It says that on average, bacterial physiology is not comparable, and you should expect specific environments to select for distinct sizes of bacteria. These are all really nice things to know ahead of time. It also tells us something about physiological flexibility, because you can see that at both the small and large end of bacteria, they're running out of space for the essential physiology. So we have this space constraint that limits now both ends of the spectrum. This happens at the same size that we saw this energetic restriction. And in between these two, you have sort of this extra space, seems to be mostly filled with water, but what comes with that is an added flexibility to, say, increase the number of a particular protein in response to environmental fluctuation. So we also now predict that there's differences in the responsiveness of bacterial physiology at different size scales, again, telling us something about what sorts of environment should select for what sorts of species. Now, everything I've said has been very much from an optimization perspective. So we've looked at, for example, the central dogma in terms of what would be optimal for cellular physiology as a function of these size shifts. But one could ask, what are the boundaries of this physiology? So it's hard to map what you would do in every particular niche, but you might get around that by just saying what are sort of how far could you push this physiology to understand its ultimate bound. And so that's sort of the hope, is how do we now start to abstract this even further to just say, even for life as we know it, what is the hard boundary around that look like for upper and lower bounds of various processes? So one way we can do that is to say, well, what happened if life had a slightly different evolutionary history? What if we discovered a ribosome that was better or worse than was discovered by the evolutionary trajectory that we had? And so you can think about the ribosome basically as the number of base pairs it can produce per second given its size. And so you get sort of this base pairs per second per volume is the critical parameter in all these models for ribosomes. And if you took the ribosome to be exactly the size as the one that we know, but you said imagine it was able to process base pairs 10 times faster or 40 times slower, well, if it could process 10 times faster, you actually get an extra order of magnitude on the upper bound for bacteria. So you should expect all bacteria to be larger. You expect to see where that transition between bacterial architecture and unicellular eukaryotic architecture to be pushed to a larger size regime. Now, it's up to the biochemist to decide if this is actually possible or not, but we can sort of, in an abstract sense, say, once we know the biochemical limit, we can back out the physiological limit. And now if the ribosome was 40 times slower, you can't get encapsulated life at all. So for all sizes, it's impossible to have enough ribosomes to actually run cellular replication. I'm holding some other features of cell physiology constant here that should be noted, so you might be able to tune those a little bit. But given that conditional statement, we can sort of understand the effect of a better or worse ribosome discovered in evolutionary history. So I think taking all of that together, what this shows is that by looking at this macrophysiology and thinking about these scaling laws, there are ways to start to go from the broad diversity of life that we've seen to some set of general principles about that life. I haven't talked much about it, but in lots of the cases where we've looked and predicted these cross-species relationships, we understand sort of the dominant physical constraint or the main physical process limiting that. So that makes those predictions very general. And then you can play the sorts of games I just described about layering the detailed physiology on top of that to understand, say, the bounds of our life. And so this gives us, hopefully, the possibility of going from physics to then a variety of physiological possibilities to understand the bounds of life or the how far we might expect something to exist away from the types of organisms that we already see. So with that, I'd like to thank a long set of collaborators, many of whom are here today. I'd like to acknowledge NASA and NSF for generous funding throughout various portions of these papers. And with that, I'd be happy to take any questions if we have time. And thank you for your attention.
So there's a microphone up front. Please use it for questions.
Hello. Okay. Hi, Mike Wong, University of Washington. I'm not really in this field, but it was a really interesting talk. And I've read in the work of like Nick Lane and others that the sort of cell size maximum can be attributed to a sort of surface area to volume ratio kind of thing where chemiosmosis happens along the surface area of cells. And so I was wondering if this ribosomal limit is complementary or contradictory to that argument, and if you have thoughts on that.
Yeah, I mean, so I won't go into that particular story in detail because we have a lot to say about that. But the broad point I will make is that many of these sort of limits occur at the same place for different perspectives. So this size-based, just this packing problem for the smallest cells, that limit is almost identical to the energetic problem. And that really tells you that two things should be being co-optimized at the same time, right? So it says that you have a set of constraints, and evolution pushes on the constraint that most matters until they've sort of all reached some boundary that they can't go beyond. And so there's some very complicated co-optimization we haven't worked out yet related to why many of these constraints occur at the same scales, and I think that relates to some of Nick Lane's arguments.
Cool, thank you.
Time for one more question.
Okay.
Hi, I'm really struggling to understand why you think cell volume is a relevant sort of independent variable to describe these processes? Because I guess thinking from my point of view as a biochemist, I mean, most bacteria are about 1 femtoliter, and that basically comprises a huge spectrum of things like E. coli that basically can double every 20 minutes and have 50,000 ribosomes, to things like soil bacteria that are the same size and basically double once every month and have maybe 1,000 ribosomes and are just as happy in terms of they can persist in their niche. So I guess if you can have like things that have such a different metabolic sort of, lifestyle, very different number of ribosomes, and are the same volume, I don't really understand why is the volume so relevant for your study?
Yeah, that's a great question. I mean, I'd say at the top level, volume's a relevant parameter, mostly because it interacts with a variety of physical constraints. So where your boundary is has dramatic implications at the, for the simplest example, for things like diffusion, right? So size immediately tells you something about the physics, and that's one of the reasons that we think about this boundary as a constraint. Now, how much you want to change that concentration within that boundary is a more complicated question. Something I should mention here too is that this is all for a maximum growth rate perspective, and under these maximum growth rate considerations, you often see shifts in cell size when you move away from those conditions. So I think there are other dimensions that you could project these relationships across. You could even do something like concentration of the ribosome as your generating parameter, and then you would see a strong correlation with cell volume, but it would decorrelate from lots of other things because it's not as directly related to certain physical constraints. So that I think it's the connection with obvious physics that makes size a natural variable. Yeah. Thank you.